In this video, we're going to make a mitosis brochure to do an overview of the purpose, the location, and the reason for mitosis, as well as the different stages that happen during mitosis. So you're going to have a green template like this. I want you to fold it in 1/3. Like this, so you have a nice brochure that can be opened, and it's also double-sided. Okay, so let's get started and work on the first page. So here's a cell cycle diagram. Let's go over the cell cycle stages. Just like humans have a life cycle, cells also have a life cycle. Their life cycle starts with G1 phase, when the cells are actively growing. Then it proceeds to S phase, that's when cells replicate its DNA. Then it goes into G2 phase, that's when cells prepare for cell division. And M phase, which stands for mitosis, this is the stage where the cell actually divides into two daughter cells. And M phase is subdivided into 5 parts. Let's zoom in. So the first part is prophase, metaphase, anaphase, telophase, followed by cytokinesis. Some textbooks can combine telophase with cytokinesis because the two happen simultaneously at the same time. And all 5 stages altogether, they are referred to as the M phase. So now let's answer these 4 questions down below. When does mitosis happen? During the M phase of cell cycle. And M phase only makes up about 5 to 10% of the entire life cycle. So mitosis is relatively a quick process. Where in the body does mitosis take place? In somatic cells. Somatic cells include all body cells, non-germline cells. So your skin cells, heart cells, liver cells, all the body cells that are not involved in reproductive functions, they undergo mitosis. Why does mitosis happen? There are 2 purposes. Number 1, for repairing damaged tissues and damaged cells. And number 2, growth. The end result of mitosis produces 2 genetically identical diploid daughter cells. We use this notation 2N for indicating diploid, and di means 2. So in diploid cells, such as your somatic cells, it contains 2 copies of each chromosome. Okay, so that's the overview of mitosis. Now we're going to get into each specific stage. We're going to draw what the cell looks like and summarize the key events. All right, so let's go into interphase. So interphase refers to the first 3 stages of the cell cycle, which includes G1, S, and G2. So let's write that down. G1, S, G2. All 3 of these stages combined, they're referred to as interphase. And that's when the cell is actively growing, duplicating its DNA, and preparing for cell division. So we're going to first draw what the cell looks like during the interphase. So this large circle represents the cell. And inside the cell, we have a nucleus, and we have a nuclear membrane surrounding the nucleus. And this nuclear membrane is intact. It's not broken down. And within the nucleus, we have the DNA. And I'm going to use 2 different colors to indicate both the maternal and the paternal copy of the chromosome that came from your mom and dad. And over in this corner, we have 2 pairs of centrioles. And centrioles are these cylindrical looking organelles that are used for cell division. And their roles will become more apparent as we proceed, as we continue in this brochure making. Okay, so now let's go ahead and summarize all the key events that happen during interphase. So as you can see, the nuclear membrane is intact. Intact, not broken down. And inside the nucleus, we have DNA in the form of chromatin. DNA is in the form chromatin, and it means that it's not condensed. Decondense. What it means is that DNA is not coiled up yet, so DNA is spread out in the nucleus and it is not compacted in any form. So if you were to look at a cell in interphase under a microscope, you won't be able to clearly see the chromosomes. And as we talked about, during the interphase, the cell grows, duplicates its DNA, and prepares for cell division. Okay, so that sums up what happens during the interphase. So now we're going to get into the actual mitotic stages. The first one is prophase. Okay, and I'm going to tell you a quick way that you can remember the key events that happen in each stage of mitosis. So remember pro for before. Prophase is the very first stage of mitosis. It happens before all the other phases. So we're going to draw what the cell looks like during prophase. And this nuclear membrane, which was intact during interphase, it now starts to break down. We're going to indicate that with dotted lines. And inside the nucleus, we have X-shaped chromosomes. They are no longer decondensed in the form of chromatin. They're now in the form of chromosome. Let's go ahead and label that. Chromosomes. And now they are visible under the microscope. So let's label this as chromatin. So these words, they both refer to DNA, just different state of DNA. Over in this corner, we still have centrioles. And these centrioles are beginning to form these rope-like structures, which we call mitotic spindles or spindle fibers. Let's label that as well. So let's summarize the key events that happen during prophase. The nuclear membrane begins to break down. And inside the nucleus, DNA is now compacted into chromosome. And again, if you were to look at a cell undergoing prophase, the X-shaped chromosomes are now clearly visible. And the centrioles are beginning to form some mitotic spindles, which will be later used to pull apart the chromosomes. Spindle fibers. Okay, now let's go over to the metaphase. I want you to remember M for middle. This is when all the chromosomes line up in the middle, ready to be separated. Okay, so let's draw what the cell looks like. The cell still looks circular. Okay, so we have centrioles kind of separated to opposite ends of the cells. And all the chromosomes are now lining up in the middle. And each chromosome is attached to spindle fibers. And they're ready to be pulled apart. So let's label some key things. So this again is still spindle fiber. And this imaginary plate at the center of the cell where all the chromosomes are lining up, we call it equatorial plate or metaphase plate. Okay, now we're ready to summarize all the key events. Number one, all chromosomes line up in the middle. In the middle of the cell, the equatorial plate. And each chromosome is attached to mitotic spindles. Is attached to spindle fibers. Okay, so that sums up what happens during the metaphase. So let's go over to the back page. Let's go over anaphase. I want you to remember A for apart. We're pulling apart the chromosomes to opposite sides. So during anaphase, the cell elongates a little bit. So it becomes longer, like so. We still have centrioles on opposite sides of the cell. And now all the chromosomes are being pulled apart to opposite sides. And each half of the chromosome is attached to spindle fibers. So this used to be one X-shaped chromosome, which is now being divided into two separate chromosomes. We call these two identical halves sister chromatids. So the sister chromatids are being pulled apart to opposite sides. And the way that they're pulled apart is the spindle fiber that's attached to each chromosome, it's actually becoming shorter. The spindle fiber is depolymerizing, it's getting shorter, and that is able to pull apart the chromosome to opposite sides. So let's go ahead and summarize the key events here. The sister chromatids are pulled apart to opposite sides. As spindle fibers depolymerize, and as we talked about it already, the cell elongates. Okay, so now let's go over to the telophase. I want you to remember T for two cells. And telophase and cytokinesis, they kind of happen at the same time. Telophase is referring to the division of the nucleus. Cytokinesis refers to the division of the cytoplasm. But they happen simultaneously at the same time. So the cell is ready to be divided into two separate cells, like so. And on each end of the cell, the nuclear membrane begins to reform. And the centrioles are also divided in half. And inside the nuclear membrane, we have those sister chromatids that have been separated already. So let's summarize the key events. So the separation of sister chromatids is completed. And the nuclear membrane begins to reform. And cytokinesis, the division of the cytoplasm, begins to proceed. To produce two daughter cells. And in this case, which I'm showing an animal cell here, this little pinching of the cell membrane that occurs, this is called cleavage furrow. So this pinching of the cytoplasm, pinching of the cell membrane occurs to divide up the cell into two daughter cells. This is what happens in animal cells. And in plant cells, we use something called cell plate. So in animal cells, cleavage furrow forms. In plant cells, cell plate forms in the middle to divide up the cytoplasm. So this sums up the stages of mitosis. So you should definitely be able to answer when, where, and why mitosis occurs, as well as the end results. And if you are given pictures or diagrams, you should be able to identify the specific stage of mitosis based on the picture. And you should be also able to describe what happens in each stage of mitosis. So that's the overview of mitosis. I hope this was helpful.